Good morning. The title of this presentation was Repetitive Transcranial Magnetic Stimulation on Dorsolateral Prefrontal Cortex Affects Cortical EEG Rhythms and Visual Episodic Memory. You can see the authors, the results of this presentation derived from the Pharmacope project granted by European Committee under the Innovative Medicine Initiative. No conflict of interest. In the Pharmacope project, we tested the biomarkers of Alzheimer's disease for drug discovery. A special interest was on biomarkers of memory functions affected in Alzheimer's disease. Ideally, these biomarkers should be able to detect brain dysfunctions even in the preclinical stages of Alzheimer's disease without cognitive deficits, and then along the disease progression to dementia. The Pharmaco Consortium included many academic and industrial partners, and you can see here the distribution in Europe of these units. The results of this presentation derive from the Work Package 1 of Pharmacope project. Focus on condition challenging the memory functions in healthy subjects for preclinical studies. And you see here the key researchers of this work package. Professor David Bartres from University of Barcelona, Professor Olivier Blanc from University of Marsella, Professor Catherine Cassé-Parot from University of Marseille, and other key researchers and industrial partners from GlaxoSmithKline and Roche. EEG rhythms are sensitive to changes in vigilance and sleep. The changes in vigilance of EEG rhythms were demonstrated by Hans Berger in 1928 with the first EEG recordings in humans. Of special interest was the alpha rhythm at about 10 Hz. The alpha rhythm is dominant in the parietal areas and occipital areas during the resting state as closed condition, and you can see ample oscillations at about 10 Hz in this condition. During awake and alertness, higher vigilance, the alpha rhythms related to cortical inhibition disappear, and you can see EEG oscillations at higher frequency in the beta range from 13 to 35 Hz and in the gamma range from 35 to 80 and higher frequencies. And then you see that EEG oscillation change in amplitude and frequency during the transition to sleep and sleep. Alpha rhythms reflect thalamocortical and cortico-thalamic synchronizing capacity of the brain. And here you see a sketch with cortical neurons and thalamic neurons from nucleus reticularis thalami and thalamic relay nucleus. And you can see the interactions between pyramidal cortical neurons and thalamic neurons and projections from thalamic neurons to cerebral cortex. It's possible to record EEG rings and alpha waves from cortical electrodes and the thalamic electrodes in animal models. And the higher the alpha REMs, the higher the cortical inhibition during wakefulness. The specific study questions of the work package 1 in this Eeg experiment were to test the hypothesis of specific EEG rings in alpha and beta frequencies related to memory retrieval of visual stimuli, specifically indoors of apartment or outdoors landscapes. Another hypothesis was that repetitive transcranial magnetic stimulation interfering with left dorsolateral prefrontal cortex may affect those rings during memory retrieval. We use strain of TMS at 20 years for short periods over left dorsolateral prefrontal cortex or vertex. You can see here the locations. The stimulation of left dorsolateral prefrontal cortex was the experimental condition in relation to previous studies of neuroimaging supporting the so-called HERA model, hemispheric encoding retrieval asymmetry model. And we validated TMS challenges over left dorsolateral prefrontal cortex according to HERA model in a previous study. In the present EEG experiment, we use the landscapes as outdoor pictures and the internals of apartment. We presented the indoors and outdoors during the encoding phase and asked subjects to respond with the left button if they perceived indoors and the right button if they perceived outdoors. After 30 minutes, we presented again some pictures shown in the encoding phase during the retrieval phase. And we added totally new pictures with indoors and outdoors. And we asked the subjects to respond pressing the left button for the old pictures presented during the encoding phase, or right button for totally new pictures. And we enrolled 16 healthy male volunteers for this experiment. Here you see an overview of the design. During the encoding phase, we gave TMS challenges over left dorsolateral prefrontal cortex with some pictures, and TMS challenge over vertex during other pictures. During the retrieval phase, we recorded EEG during the presentation of old pictures presented in the encoding phase and totally new pictures. The EEG period of interest was in the first second of the presentation, and we used 19 scalp electrodes according to 1020 electrode system for this exploratory study with low EEG resolution. The artifact-free EEG data were used as an input for the estimation of cortical sources of EEG activity by exact low rate of freeware, which is a freeware that all people can download from internet. Due to the low resolution of EEG recordings, we used large cortical regions of interest for the source estimation. And you see a large frontal cortical region of interest, a central region, and a parietal region. Here you see again the design with the encoding phase and TMS challenge over left dorsolateral prefrontal cortex in some pictures, or vertex TMS challenge in other pictures. The EEG recording during the retrieval, and for the retrieval phase we computed the accuracy in the detection of totally new pictures presented only during the retrieval phase, or old pictures already presented during the encoding phase during the vertex challenges with TMS, or with the TMS challenge of dorsolateral prefrontal cortex in the left hemisphere. And you can see that the detection of the new pictures was high, about 90% of accuracy. The correct detection of old pictures receiving vertex TMS during the encoding phase were lower, 75%, and ever lower was the recognition, the correct recognition of old pictures receiving the TMS challenge over dorsolateral prefrontal cortex in the left hemisphere during the encoding phase, only 62%. So there was a clear effect of TMS over left dorsolateral prefrontal cortex during the encoding phase on the pictures in relation to retrieval. And we replicated the results of previous studies. The new findings regarded the EEG. Here you see again the design with the encoding phase and TMS challenges over frontal areas and the vertex, and the retrieval phase with the EEG recordings. You see here the alpha reduction of power, or alpha event-related desynchronization, ERD. The percentage reduction of alpha reflects the cortical activation, and you see a clear cortical activation as revealed by alpha ERD in parietal areas and central areas. Interestingly, there were no significant differences between alpha ERD cortical activation for the totally new pictures in the retrieval phase, or old pictures receiving TMS challenges in the vertex and or left dorsolateral prefrontal cortex. Similar cortical activation means that alpha RINs are sensitive to cortical arousal in general for cognitive processing or complex visual stimuli, but not specific memory processes for old pictures to be recognized. In contrast, beta RINs show reduction in power, or beta ERD, with some differences between conditions. You see that during the retrieval phase, the totally new pictures were related to a cortical activation as revealed by beta power reduction, or ERD in blue. But even more reduction in power of beta rins and cortical activation during the recognition of pictures, old pictures, receiving TMS challenge over vertex or TMS challenge over left dorsolateral prefrontal cortex. So beta rims are sensitive to the recognition of old pictures compared to new pictures. So they are sensitive to specific episodic memory processes in the retrieval phase. Furthermore, you can see a slight but interesting difference between the cortical activation beta ERD during old pictures receiving vertex TMS challenge and left dorsolateral prefrontal cortex. It's a slight but interesting difference, showing that old pictures receiving left dorsolateral prefrontal cortex challenges during encoding phase are related to less cortical activation and maybe less recognition accuracy during the retrieval phase. In conclusion, alpha reduction in power, or ERD, may reflect widespread cortical arousal related to both detection of old pictures presented in the encoding phase or totally new pictures, but not sensitive to TMS challenge during the encoding phase. In contrast, beta reads the reduction of power, ERD, may reflect frontoparietal activity related to the detection of old pictures, so specific for episodic memory processes, and may be slightly affected by frontal TMS challenge during the encoding phase. This finding needs more studies with high-resolution EEG system, allowing more circumscribed areas in the source space and more subjects to increase the statistical power. Thanks for your attention.